Hello, everyone. In this infographic media, we are going to discuss the growth, regeneration, and evolution of organisms. Join us as we discuss the main concepts of this chapter, including tissue organization and stem cells, growth, aging, and cancer, regeneration of missing parts, and evolution and development.
The development of tissues occurs with the continuous production of new differentiated cells from populations of stem cells. Let us first talk about the primary tissue types found in the vertebrate body and the various stem cells found in the organs that renew the cells of these tissues. The organs of the body are mostly composed of more than one tissue layer. A tissue is a set of cell types formed from a particular type of progenitor cell or stem cell. An organ or body part normally contains several tissue types arranged to fulfill a common function, and these are usually derived from more than one embryonic origin. Traditional histology distinguishes various types of tissue. The epithelia is a sheet of cells arranged on a basement membrane with each cell joined to its neighbors by specialized junctions. Epithelia may be simple, stratified, or pseudostratified, and they may also be squamous and glandular. Connective tissues usually refers to those tissues dominated by fibroblasts, such as the dermis of the skin and the fibrous capsule surrounding most organs. The types of connective tissue include the skeletal tissues composed of cartilage and bone, and the adipose tissues. Muscle tissue is a specialized tissue found in animals which functions by contracting, thereby applying forces to different parts of the body. Muscle tissue consists of fibers of muscle cells connected together in sheets and fibers. The types of muscle tissue include striated, smooth, and the cardiac muscle. The blood is composed of myeloid cells and lymphocytes, which arise from the hematopoietic stem cells in the bone marrow. Furthermore, the blood vessels have three layers composed of endothelial cells, smooth muscle, and fibrous connective tissues. Stem cells are cells that can both renew themselves and generate differentiated progeny. Stem cells can be embryonic, which are pluripotent stem cells, or they can be tissue-specific stem cells committed to form the cell types of a specific tissue. In this video, we will deal more with tissue-specific stem cells. Tissue-specific stem cells are visibly undifferentiated and whose progeny include both further stem cells and cells destined to differentiate. They persist for the lifetime of the animal and are found in niches defined by signals from surrounding cells. Tissue-specific stem cells are committed to form just one particular tissue type, and they are found in the different organs of the body. For the small intestine, it has its proliferative zone in the lower part of the crypts. Intestinal stem cells are found adjacent to the Paneth cells at the crypt base. The epithelium has proliferative cells only in its basal layer, and the successive higher layers represent degrees of post-mitotic cell maturation. The epithelial stem cells are located in the basal layer. The hair follicles contain stem cells in the bulge of the outer root sheath that reconstitute the hair bulb in each active phase of the hair cycle. The bone marrow contains hematopoietic stem cells, which renew the cells of the blood and immune system. Lastly, spermatogonial stem cells adjacent to Sertoli cells in the tubules of the testes produce sperm continuously in male mammals.
Biological development is the progressive changes in size, shape, and function during the life of an organism. In this part of the video, we will talk about growth, aging, and cancer. Although superficially familiar to all, growth is one of the less well-understood aspects of development. By the time the general body plan is formed, the whole embryo will be about 1 mm long and each organ is present as a small rudiment. In large animals such as the human, there will be an approximately 10 to the 9th-fold increase in volume from this stage to that of the full-grown adult. This enormous change is driven largely by cell division, but also by the secretion of extracellular matrix and by some increase in cell size. However, the overall control of growth is still somewhat mysterious. One problem relates to final size, or why the animal stops growing at a particular point, and the other main issue is the coordination of growth between parts of the body. Another process of development is aging. However, there has long been a debate about whether aging is really a developmental process or not. The argument in favor is that senescence and death arise from pre-programmed events, which are under developmental control. The argument against is that such events are simply due to progressive mutation and other damage, which have an inherently random character. In fact, both positions have proved correct to some extent. Aging is developmental insofar as it responds to genes that can be manipulated, and remarkably, it turns out that one of the important systems controlling it is the same as that regulating growth, namely the insulin signaling pathway. One example of growth that is detrimental to the human body in particular is cancer. Cancer is of huge interest to human beings since about 40% of us will get some form of cancer, and about half this number will die from it. Cancer is fundamentally a growth of body's own tissue to an inappropriate extent or in an inappropriate place. But it is not the only type of inappropriate postnatal development. There are a variety of pathological abnormalities of normal growth and differentiation, classified as hyperplasia, dysplasia, metaplasia, and neoplasia. Neoplasia can be identified as benign or malignant. The distinction between the two mostly depends on their propensity to invade surrounding tissues and establish metastasis elsewhere in the body. Cancers retain many of the properties of their parent tissue, but change properties as the disease progresses. Development of a cancer normally requires several mutational changes to occur in a clone derived from a single cell. Key steps are gain-of-function mutations of oncogenes, loss of tumor suppressor genes, invasion of apoptosis, regulation of telomerase, promotion of angiogenesis, and changes leading to metastatic spread.
Regeneration of missing parts. Regeneration is the natural process of replacing or restoring damaged or missing cells, tissues, organs, or even an entire body part to full function in plants and animals. Scientists are studying regeneration for its potential uses in medicine, such as treating a variety of injuries and diseases. To start off, the regeneration of the muscle. We have two types of regeneration discussed, namely the regenerative regeneration and the restorative regeneration. The difference is that regenerative regeneration is about reconstructing a new body part or the one lost by the organism. Meanwhile, the restorative regeneration is about bringing back or stitching back the original part. The planarian regeneration, a little background. Planarians belong to phylum Platyhelminthes, and their mechanism constitutes of having cell turnovers and neoblasts. Their regeneration ability is that they can regenerate a new tail region and a new head region if cut in half, and their blastema enlarges by cell recruitment and redifferentiates over a few days to form the missing structures. As for the insects, in insect limb regeneration, these are mainly observed in crickets and Drosophila. It has a 4-step process, starting with the amputation, proceeding with the scab formation, next is the epidermis over scab, and lastly is the occurrence of blastema. And for the vertebrate limb regeneration, it occurs 2 to 3 weeks in larvae and a few months in adults. Factors that affect the vertebrate regeneration are Hox genes, fibroblast growth factors, and sonnet hedgehog. Its source cells are called progenitor cells, which are the local to the amputation surface and are formed by dedifferentiation of functional cells. Neurotrophic factor are mitogenic factors released by the nerves that is required for the growth of the blastema.
Finally, let us talk about the last part of this chapter, which is on evolution and development. Join me as we go through all the important key points to remember in this topic. First, evolution involves both adaptive changes brought about by natural selection and neutral changes established by genetic drift. Neutral changes are most apparent in DNA and protein sequences. However, the importance in evolution in the issue of developmental constraints and of macro and micro mutations is still not known. Next, classification of living organisms usually attempts to represent the evolutionary history of the groups concerned. Now, this is called a phylogenetic or cladistic taxonomy. Homology between the parts of different animals was traditionally determined by methods of comparative anatomy, including a study of developmental stages. Homologous features are similar because they derive from a common ancestor. Analogous features are similar because they are driven together by natural selection. To further represent the evolutionary relationships among organisms, a phylogenetic tree for the animal phyla themselves can be created using information from descriptive embryology, molecular taxonomy, and the fossil record. It is thought that the common ancestor of all animals lived between 1,000 and 600 million years ago. The time calibration for phylogenetic trees comes ultimately from the radioactive dating of sedimentary rocks. This provides absolute dates for divergence times between lineages, which are observed in the fossil record. Moving on, animal taxa often have a phylotypic stage at which all groups show maximum similarity. All triploblastic animals or the bilateria share expression domains and function of a basic set of genes responsible for setting up the general body plan. This configuration of gene activity comprises the HOX, para-HOX, and NK clusters, and also a certain other genes including OTX, EMX, and PAX6. This configuration, or the zootype, was probably present in the common ancestor of all triploblastic animals. Going further onto different animal groups, in arthropods, changes in their segmental organization have arisen partly from changes in HOX expression, but mostly from changes in downstream genes controlled by the HOX pattern. Also, in tetrapods, the basic developmental mechanisms of tetrapod limb development exist and are also observed in the paired fins of fishes.
To sum it up, growth and development in animals is a very complex process. Indeed, all the essential ideas and key points in developmental biology play an important role in understanding these mechanisms. We hope you learned a lot in this presentation. Thank you for watching!